Well, welcome everybody. I'd love to introduce Hani Goodarzi from the Arc Institute and UC San Francisco, UCSF, who's been focusing on modeling and ML techniques for analyzing the cell transcriptome. Welcome, Hani.
Thank you, Greg, for both having me and for the introduction. I'm excited to be here and tell you a little bit about the work that we have been doing at the intersection of AI and biology. That's kind of where we operate. This is one example of that. It's a work that we just recently pre-printed. So as I'll refer in the end as well. It's a good time to talk about it. Well, just to reiterate, I'm Hani. I hail from UCSF, and as of last year, I'm a core investigator at Arc Institute. I also lead the Computational Technology Center here. Actually, just recruited Dave Burke out of Google to become our CTO. So he'll take over some of that, operational aspects of it, and I can go back to just doing science. Yeah, excited to tell you a bit about what the work that we have been doing here at ARC, kind of basically modeling the human transcriptome. And if you don't know what transcriptome means, I'll explain just now. What is transcriptome architecture? So basically every gene in our body, every cell in our body has the same 20,000 or so genes. These are the most common genes, but the genes are just not a very clear end-to-end, like this is the beginning, this is the end of what they actually code. In reality, they're partitioned into what we call exons, and what goes in the middle is introns. And one of the ways that we get a lot of complexity out of the genome is basically alternative splicing, which is how these exons are then put together to actually create variations of coding sequences. And so the resulting proteins that comes from a gene can be actually different depending on which exons are included or excluded. And as a result of it, they can actually carry out different functions. So these are called isoforms at the RNA level. So the same gene can lead to many isoforms. Not only can, it often does, even some of them going to hundreds of different isoforms. And each of these isoforms have a different abundance in the cell. And actually, being able to quantify these isoforms is a very important important problem in biology, as I'll talk about. But basically, what we define as transcriptome architecture is the makeup of these isoforms for every gene and their individual abundances. Okay. So as I mentioned, this is kind of the makeup of one event. So there's one exon on this end and one exon on the other end, and then this piece in the middle is called an intron, and this will get removed. So this is a very common machinery in the cell. It's called the splicing machinery that is in charge of this. And there is an entire cis-regulatory grammar sitting on top of this machinery that guides like how it should splice things. But at the core of it, the job of this machinery is to just remove the introns and splice together these exons into a contiguous coding sequence. Okay, so understanding how this process is regulated is not a new problem in biology. It has been around for many years. So we're really standing on the shoulders of giants, as one would say. There are, basically a variety of different modeling approaches over the years that have under the category of like sequence to function mapping, machine learning, that have really tried to tackle this problem as well, basically learning the relationship between sequence and splicing. And I really should emphasize that the point is that, once you have this RNA, which is, this is called a pre-mRNA, which is the exact copy of the DNA itself. So this pre-mRNA is the exact sequence is the RNA form, and then it gets spliced. So much of the information, if not all of it, in terms of how this pre-mRNA gets spliced, should really be already encoded into that RNA, right? So there should be basically barcodes in different parts of this RNA, which we call cis-regulatory elements, basically elements in the RNA itself that act on cis mode on the RNA itself. That is basically what we call splicing grammar. Basically, what are these regulatory elements in the RNA that define how it gets processed in the cell? So this is basically called the splicing code. in biological lingo. And basically learning the relationship between sequence and splicing is basically deciphering this splicing code. And going back more than 10 years ago, for example, Brendan Frey's group at University of Toronto, he published a seminal paper in this called splice code, and that was actually one of the foundational models that went into the company called Deep Genomics. More recently, for example, Splice AI was published, where you have basically a more recent convolutional model where it's a deep convolutional model that basically predicts the splicing events as well. So you have a span of, these models are apart, 5 years or so. But and there have been a lot of progress, but it's still one of the biggest challenges that remains with these models is context. So even the most recent models like Splice AI, they actually have about 10K context around splice sites. However, the reality is that genes are much, much longer than that, especially introns are very long. So the vast majority of our genes actually fall above like 50K length. So that's kind of a context that you need to be at to be able to model the entirety of the gene in one input, in one go. Of course, along these lines, there have also been RNA foundation models. Basically, as everyone knows what they are at this point, they're basically large language models that are pre-trained on the genome and the transcriptome. There are a variety of them. For example, Calico, David Kelly has a series of models like Borzoi and Reformer that basically model the genome to transcriptome relationship, not specifically splicing, but there are things in the splicing that the model understands. There's big RNA out of Deep Genomics, again, Brendan Frey's group. There's also like other models like nucleotide transformer and Hyena DNA that they're again genome models, but they can capture aspects of the transcriptome architecture as well. But really a fully customized understanding of transcriptome and transcriptome architecture is generally lacking, especially at nucleotide resolution with its full context. So for example, a lot of these models, they achieve the context that they do, like, big RNA or Borzoi, by combining convolution and attention. So they're not like nucleotide resolution models. Splicing code is nucleotide resolution. So you need to be able to really capture that as lowest resolution, as highest resolution as possible. So this is the
So,
statement of the problem. Go ahead, Greg.
also maybe this is what about, I'm trying to understand the statement of the underlying biology, but here's what it sounds like to me. You have a sequence of exons, and you have to kind of identify what the introns truly are, remove them, and they could be arbitrarily long, but then different subsequences of exons form different proteins.
Yeah, exactly. So depending on which introns you remove. So for example, let's say this is a gene. As you see, it has non-coding regions that are part of the exon. So this is exon 1, this is exon 2, exon 3, and exon 4. And there are also 3 introns in the middle. So if you remove all of these introns one after another, you get a nice 1, 2, 3, 4 mRNA. But you could also remove the beginning of this intron and end of that intron. So you get an RNA that actually doesn't have exon 2 even, right?
Good.
And
So
then
let me
there,
get this. Do you have a clear rule for what an intron is, or is that ambiguous as well?
uh, well, when I talk about the data, you will see like we're starting it as an input to the model, so we know what is an intron and what an exon is. The question is more, how are they spliced together?
Got it.
Right?
Go on. Thank you.
Because as I said, for example, the beginning and end of introns can mix and match. So for example, you can start here and end there, or you can start here and end there, or you can start here and there. So you can actually, I don't actually know if you see my mouse or not. If
I
I'm
do not, but this makes sense.
okay,
And
so
then,
maybe, how about
oh,
now?
yes, now I see it. Thank you.
Okay, so you can start here and end here. That could be your intron. Or you can start here and end there. So that can also be an intron basically when it gets removed. But that step will actually remove exon 2. So the resulting RNA that comes out of that will not have exon 2 anymore. And the protein will lack the part that is coded by exon 2.
So, okay, you can have arbitrarily long skips. Well, first of all,
Yeah.
arbitrarily long, or they're reasonable bounds for how much you can skip.
I mean, they're like it's rare from end to end. It's mostly local. However, there are also longer interactions. So, for example, if you miss exon 2, maybe you're also missing exon 5. So there's this kind of longer interaction, which is why we need a long context. So the isoforms that have this kind of longer interactions, that there is this tie between different exons. So whenever exon 2 is there, exon 5 is also there. But how
Great.
is that accomplished, right? So that's exactly what the splicing code is.
And our
So for
viewer,
example, like exon, it's either exon 2 there or exon 3 there, never together. So this is a mutually exclusive exon mapping. So these are the kind of different versions of how a splicing happens that gives rise to all of these different isoforms, which in turn give to different versions of proteins from the same gene.
and our reordering is possible, or is it always in this sequential order?
It's always in a sequential.
Got it. Thank you.
Yeah, I mean, it's very rare that you will get back splicing and that results in a circular RNA, but that goes way into the biology. For all intents and purposes, this is a nicely ordered process.
Got it. Thank you.
Okay. So basically, this is the statement of the problem. So our goal is to build an RNA foundation model that can represent the entirety of the context of the gene, so capture the entirety of the gene, and be able to understand how this gene will then be spliced into different isoforms. Okay. And we want to do this as nucleotide resolution, basically. So we need two things to be able to do this. Of course, one is the training data and one is the model. I mean, one thing that you probably have noticed is that, you probably have, when it comes to machine learning in biology, you probably have heard a ton about, protein models, right? Protein language models. You've probably heard a ton about genome models, genome foundation models, including EVO, which folks at ARC train. Um, but you hear less about RNA, and partly because RNA models are just data starved. So there isn't as much data out there, relevant data for foundation modeling. I mean, it depends also on the foundation model, but for this kind of foundation modeling, there isn't just simply not enough data out there to be able to train a large language model. So the first step to solve this problem is also thinking through what a comprehensive database of isoform architectures would look like. And the second part of it is also model building that I will talk about. So let's start with training it. So why isn't there enough data out there? The reason that there isn't enough data out there is that much of the data in genomics is generated through what is called short read sequencing. So where the longer, basically these are your different isoforms. Again, you have one gene and then you have all these different isoforms, right? It begins and ends different places. The skips happen in different exons. So you give all of these different variants of the same gene. And but you traditionally you do not actually sequence end to end these RNAs. You just fragment them into small pieces, and then you sequence those fragments using more commonly than other platforms, Illumina sequencing. So you basically can decode these like fragments that here are shown as these like green fragments, and every now and then you fall basically on a junction like these red fragments that there's a skip. You actually see the skip, right? So you get the picture of how things are spliced, but a very local picture, right? So for example, you see here that there is a skip here, but you don't know that how this skip is then related to all the other skips. Are they on the same transcript? Are they not? You don't have the global understanding of how the isoform looks. You just have the very much small and like local understanding of how splicing can happen at each of these events, but not how they line up together. And there has been a ton of work trying to go from this kind of shorter fragmentations to deducing what these longer form isoforms will actually be, right? But then in more recent years, we have had, thanks to advances in genomic technologies, now we have tools to sequence RNA actually end to end, right? So one of the technologies that we use is called it's an instrument called Sequel2, who's uh, designed and sold by PacBio. Pacific Biosciences. They've been around for a long time, but now they have an instrument that can actually do this at high enough throughput. But because these instruments are new and often expensive to run, there simply hasn't been enough data of like this magnitude of end-to-end RNA that you can actually take advantage of to really map individual isoforms. So this is the problem that we set out to solve. So we created this compendium that we call Cancer Cell Line Long Read Compendium. We bought a Sequel2 instrument a long time ago, and we have been putting it to good use. Basically looking at the transcriptome, basically for cells from different organs. These are all like cancer cell line models, so they are coming from different tumors, but tumors from different tissues of origin. So from skin to lung, to breast, to liver, and so on and so forth. And for each of these cells, we have both the long read, like the long read full transcriptome sequencing, and also the short read sequencing. So why do we need both? We need both because these are actually, like the throughput is actually a lot higher in the short read. So if you really want to quantify the isoforms really well, you actually need to have short read data as well. Because here you are in the regime of like, oh, 1, 2, 3, 4, 5 reads per isoform. So it's like very, very low counts and subject to noise. Whereas given this, once you have short read data, you're actually very good at post hoc assigning different fragments back to the full isoform and be able to quantify each of these isoforms. And this is work that others have done as well over the years, including us. But basically, this gives you like how many, like what the read length is. You can see we are sequencing on average, reads about 3,500 long RNAs. In comparison, the short read sequencing usually gives you on average 400 base pair long sequences. So this is an order of magnitude longer sequences. That's why this is useful. And you also can see like kind of like library sizes, the biotypes of the RNAs that are read out. One thing I will point out here is that the green bars here are isoforms that were previously annotated. And the rest are actually novel. So, we have found basically new isoforms that hadn't been seen before, and we have found them because we have done this end-to-end sequencing, if you will. So as you can see, there's a good fraction in every cell line that's actually new. Okay, so we started with this C2L2 project, which is 52 samples. There are 2 replicates for 26 cell lines. We also included as much other data as we had processed over the years. So we had done a total of 149 human samples and 46 mouse samples, and from a variety of tissues and cancers and normal and even some COVID samples in there. So in total, we had 2 species, close to 250 samples, close to 100 million full length reads, about like 500,000 unique isoforms. So it's a very large number. And in terms of, again, here you can see the number of samples for human and mouse. So we have 2 species, of course, a lot more in human, but we also have mouse as well. Okay, so this is our training data. All in all, we have about, counting every nucleotide, as I'll explain what this means, but counting at every nucleotide as a token, we have about 7 billion training tokens here to be able to train a model. So if there are no questions so far, I can talk about the model. Excellent. So the model that we have built on this data, we have called it LoRNA-SH, basically long RNA model. And SH stands for StripedHyena, which is the architecture that I'll talk about. But basically, our model is called LoRNA. That's what we call it for short. As far as we know, it's the first RNA foundation model that is specifically designed and trained on the transcriptome architecture, basically capturing the entirety of the context of the gene. So it's a long context model. And basically, it captures as input the entirety of the gene from the beginning to end, and all the isoforms fit within that context. But in there, you have, if you take an isoform, you have DNA parts, because the gene will start from the first isoform, where the first isoform starts. But isoforms can start from different places. So some isoforms can start further into the gene. Same at the other end. Some isoforms end further along than others. So the ending of isoforms can also be different. And in the middle, they can also have different introns and different exons makeup of those. So you have, if you take the entirety of the gene, there are, for every isoform, there are parts of it that are going to be DNA content because the transcript hasn't even started yet or has already ended. That you have exonic content, which are these boxes, and then you have intronic content, which is basically these lines. So
Actually, let me clarify what you mean
sure.
here. So when you say DNA content, do you mean that you scan DNA separately and you know that it starts at this part of the DNA, but those those base pairs are not part of the mRNA that you scan in this experiment?
Yeah. So this is an example, right? So this gene starts here
Mhm.
and ends here. Okay. But this isoform starts here. So it starts way inside the gene, right?
Mhm.
So it starts here, ends there. So it ends where everything else ends, but it starts into the gene. So when we give the gene as an input, this part is not considered part of the transcript. So it's really the DNA part. It's not the RNA part.
Cool. And then
That's
do you provide the preceding DNA part in your model?
exactly. For everything, you get the entirety of the gene, so from the beginning to the end as the input, and then you demarcate what parts is what,
Okay,
basically.
so you have to have pre-aligned the mRNA against the DNA and have marked these
Yes,
points where
exactly.
it's missing, present, and so. Okay, thank you.
Exactly. And this is exactly how we tokenize it. So we have WXYZ for ACTG in DNA. So basically we tokenize the DNA part with these tokens. And then we have a special token called S, which is the start of the transcript. And then it's the RNA part, which is ACUG. You have capital letters tokens for exons and small tokens for introns. So this is how the exons and intron tokens are described differently. And then there is again a special character token called P, which is the end of the transcript, and the rest again are DNA parts. So WZX, so on and so forth. So there is also another token that everything is prepended by, which is the species. So either human or mouse. So with all of these tokens, we basically capture the entirety of the gene and different isoforms, basically. So for example, once this will be input to the model, once this would be the input to the model. So they're the same gene, but then they're tokenized differently because this here will become an intron, and then you will get intronic tokens as opposed to exonic tokens. As I mentioned, the length of the input is about 64K. So the vast majority of genes will fit within this window. So again, we have this long context model. It can fit everything. The way we train it is basically causal language modeling. So it's next token prediction. So we start from one end, and then we just predict what comes next, um, teacher reinforcement, all the end until we get to the end of the gene, basically. And so at every position, you have to predict whether the transcript has started or not, whether you're an exon or intron, or the transcript has ended, and so on and so forth. So basically, this is what architecture means, right? You're capturing every piece of this gene for this given isoform using this tokenization. Now, what sits in the middle, as I mentioned, is a StripedHyena model, which is a combination of Hyena operator layers and attention layers. Um, and so basically, we have subquadratic, we have like subquadratic growth as opposed to attention. So this is what allows us to go to 64K. So again, the modeling, the training is basically causal language modeling. So at every point in time, you have what came before, and for that position, you basically predict what should go next. So next token prediction, and you have all of these different options. It's basically a classification problem. So you have cross entropy loss, and you also have perplexity as well as a model performance evaluation. So first, let's talk about scaling laws. I mean, there is this notion that the larger the model, the better, and that often holds true to some extent. Uh, More training flops, the better. That also holds true to some extent. But at some point, you hit diminishing returns, and the performance deteriorates from there. So as you can see, so here that those criteria. So as we increase flops, we can get better and better. In terms of this is perplexity, so lower is better. So as you can see, this movement from lower flops to higher flops towards shifting towards lower perplexities. The size of the model also is important. The larger the model, the better. But at some point, you start to overfit, so the performance deteriorates. So this is basically our leading edge. So this is the model that we picked as our basically best model to train. Again, 16 layers, 13 hyenas, and 3 attentions, 256 latent dimensions, and 7 million parameters total. Now, also to kind of emphasize the different parts of this architecture. So as I showed you earlier, there are hyena layers interspersed by attention. So if you remove, so this is what happens if you remove the hyena layer. So you only have basically a few attention layers. So the model doesn't really do too well. This is when you keep the hyena layers but remove the attention layers, and this is the StripedHyena as a whole. So you can see the StripedHyena model does performs best.
So, question.
Sure.
So, I personally am not familiar with hyena layers, but maybe what's the intuition for what dynamics of the transcription are captured? Like, why this design? What phenomena of transcription are you trying to capture using this architecture?
So I mean, there really isn't there isn't enough time to go into, but Hyena. But basically, Hyena is a space state model, and I've actually spelled it wrong here. It's Hyena. The point is that it's subquadratic, unlike attention. So attention is quadratic growth. So with attention alone, you cannot go 64K. It's a massive KQV kind of calculations around 64K that you can basically do. So Hyena is basically Hyena operators solve that problem. So there are attention-like operators, but they're space state models that capture the same relationship of long-term, long context relationships without the quadratic growth.
Okay, so the intuition should be, I mean, it sounds like we should still be thinking of like a sentence. You're trying to go arbitrarily far earlier or possibly later to have dependencies. I mean, you're still thinking like a linear relationship at arbitrary distances.
Yeah, absolutely. Because sometimes like to know where a transcript ends, you need to know where it started. So you have these like very long context interactions in isoforms that requires that kind of length, and attention is just grows too quickly.
Okay, and then from what you were saying earlier is there are some complex dependency relationships. Is that, and presumably this would allow you to learn those. Were you, is it possible to get those back to understand those interactions between different regions of the transcriptome or the process?
Yes, I'll talk about some of that, but absolutely, that's exactly what you can do, and that's why you would need, yeah, it's one of the reasons you need a model like this.
Right. Okay, thank you.
Okay, so yeah, now let's talk about that. So we have this model. It does all of this. Apparently it can predict next tokens, right? It's the foundation model. So is this useful at all? And how do we actually determine if it's useful? So as all language models out there, you can basically ask this question in a variety of ways. One is like just downstream tasks, zero shot or few shot learning tasks. That's one side. The other side of it is basically model dissection interpretation. Like what is it? An example of what you just said. Like what is it that the model has actually learned? So this is basically a causal language modeling. So it basically gives you a series of likelihoods, and you can basically calculate the likelihood of an isoform given the input, right? So for example, we take the end of the isoform, which is P here. So the output of the model for this position is basically the likelihood of seeing P in this position given the rest of the context. So it's basically that simple. And then basically from one end to the other, like you basically have like the likelihood of isoform, right, as a sum of all these pieces. Okay. Now, how is this useful, right? So it turns out what we define as a likelihood of the model, right? So here, for example, we have 3 different isoforms. We can give this as input to the model. So the model gives us 3 likelihoods, 3 scores for how likely this isoform is based on model's understanding of the entirety of the context. And then we compared this to the abundance of these isoforms. So, we had done actual sequencing. We know there's like true abundance of these isoforms. So what we showed the model was the isoform, nothing about their abundance. So it's just the structure of the isoform, not how common it is, how well expressed it is in the cell. But we do have that information. So it turns out these two are actually quite correlated. So we see that there is a nice correlation in our model between the likelihood of each isoform and the abundance and the real world abundance of that isoform. So you see a Pearson correlation about 0.15 for across all transcripts, all isoforms. And if you look at Hyena DNA or nucleotide transformer, which are 2 genome models, they have no understanding of this. Right? And we picked only these 2 models because these are the only models that are long context enough to actually do this task, where you can have a 64 kb input. We can also do this correlation gene by gene. So for every gene, ask how is the likelihood of isoforms of that gene correlated with the expression of isoforms of that gene. And so you get a distribution of values. Again, our model does a pretty good job of capturing that even gene by gene, going from an average of, again, about 0.2 to 0.5. Again, the other 2 models don't do as well, right, the genome models. So the point is that this is like biologically very intuitive, right? A splicing pattern that is more likely means that it happens more readily in the cell, which means that you will just see it more often and you will get higher abundance. That's basically the idea. Okay.
Well,
So this kind of
question.
a sure.
The impression is, so one thing I'm surprised by this because this is the transcription, not a live cell. But on the other hand, my intuition that I'm getting from you is you've biased your data set. You have a few particular cell lines where the transcription was at a particular rate because the cell died whenever you shoved it in, whenever you started collecting it, and you're actually capturing the state of the cell at that transcription state, and that's
Yeah.
what you
So
said now.
yeah, so the abundance is actually average of everything. So we have like many samples, like 26 different cell lines. What we have defined as abundance here is the average of all of those. So there's some variation depending on the context, and that context we haven't really captured. But by and large, while there are differences between cell lines, the vast majority of the numbers are also consistent. So the absolute correlation of transcript abundances, isoform abundances between cell lines is very high, which is why you can just use average as like a universal measure of how abundant every isoform is.
And it sounds like if you put in cell lines as additional tokens, or perhaps even time of day, or just additional information about what was happening when you harvested the cells, you can get this probability up, like the context, you can get the probabilities up.
Absolutely, you can condition this, like
Mhm.
you can have a special tokens for different cell lines. Just that you need a lot more data. It's
Of
more
course.
you get more data limited basically, right? So
Interesting
for a
point.
cell type, you just need to go and like get a lot more samples to be able to do this. Right?
Okay.
So that was one example of like a zero-shot task, right? Like how can we predict basically without anything, just using the model likelihood outputs um, as as a way of estimating what the transcript abundances would be without any new training. So that's zero-shot prediction task that was interesting. Another useful data set that was recently published was basically this data set that came out of University of Toronto, where they basically created like a DNA themselves, where they have two exons. These two exons are fixed, and there's an intron in the middle, and they put random fragments in the middle, right? And then these random fragments will be, if they act as exons, they will get spliced in, so you will get a new exon in the middle. And if they don't, then you will not. So this is called this is a data set called exon trapping. So this is your way of like trying to find exons by putting random fragments in there, and if you find an exon, it will be spliced into the construct, and so you will see an exon in the middle. But if it doesn't act as an exon, which are all these blue fragments, they will just get spliced out and you won't see anything. So there's this basically exon trapping for new exons, right? Because none of these exons were in our training data, right? So these are like a new data set of exons that have never existed. So this is why it's a zero-shot task. So we took this data set and we evaluated it. So how do you do this? Again, now you have the backbone. The backbone is constant, but you're putting something in the middle, and that what you're putting in could be an exon or may not be an exon, right? So you get two isoforms. So either you use the exon tokens for the piece that you put in, or you use the intron tokens, and then you get a likelihood between these two, a delta likelihood. Right? So the idea is that if this is more likely than that, then it means that it could be an exon, and the opposite means that it's probably not, right? But the nice thing is that, again, in this case, we have experimentally verified these numbers, right? So here, for example, you see that the delta likelihood is only 0.2, whereas here is the delta likelihood is 0.7. So the model thinks that this is an exon, like with a likelihood of 0.8, this piece that we put in is an exon. And you see that also more with a higher count in the data than you will see the other one. So this is another zero-shot task where we are showing entirely new exons to the model and asking whether the model understands that these are exons. And of course, some of them are also not exons. So it's both sides, right? But here, this is basically a continuous count. So going from 0, if something is never an exon, number will be 0, to thousands for something that is more commonly an exon. And again, our model zero-shot can do a pretty good job of capturing this. Right? So you see a Pearson correlation of about 0.2 for our model. The other models don't do as well. In this case, this gene is small enough that we could actually include a splice AI as well, which is one of the models I would consider the kind of state of the art before our model. And as you can see, we do quite a bit better at identifying these novel exons than a splice AI. So
So when
these
you
are
say, question, when you
sure,
say novel exons, these are gene sequences that have not been in your data set, and you're saying this is a
exactly.
plausible sequence to be included. So is it model capturing like plausible protein shapes?
The model is basically capturing this isoform is completely synthetic. Has
Yeah.
never existed. So this gene is not a real gene; it's a design gene, right? So what the experimental data captures is basically just these exons that within this context will work as exons, or rather how well they will work as exons, right? So the entire problem is a completely synthetic data set of isoforms.
So now,
Okay,
what does it mean for it to work as an exon? Like, can create a
so
protein that folds sanely? Like, I'm trying to figure out
it's
what's
not
the
about protein; it's
reality.
about splicing. So
Right. Yeah.
if this acts as an exon, the exon will be spliced into here, and you will basically get this is an example, right? So you see that the red part is an exon, so you will get it in the RNA. This is all of it at the RNA level. If it doesn't act as an exon, you will not get the red box in here. You will just get the gray parts.
So what I'm trying to understand is what makes something a good exon if it's not.
Its ability to be spliced into here.
So it signals something to the splicing mechanisms, or if it is included, the protein wobbles reasonably.
It has nothing to do with protein. Um, we'll talk about what the signals are for
Mm,
splicing.
that's okay.
Whatever they have, it's great, right? It works great, and they will be included as an exon into the isoform. And if they don't have whatever it takes to make a good exon, they won't be.
Okay.
So they will be considered an intron, right?
Got it. Okay.
So the point of this data set is that it creates an entire synthetic data that's outside of the universe of endogenous, like natural sequences that we have trained on. Uh, so it allows our model to basically flux out of the distribution of endogenous sequences and ask, how would they do in a case where we have experimental data. Again, our model zero shot can predict that quite well.
Cool.
And finally, another data set that we can use for zero shot as a zero shot task is pathogenic variations. So, you all know about genetics. We all have individual differences in our genome, and some of these differences cause diseases. Some of them are obvious as to why. So they tend to be in the coding sequence, so they will impact the protein. So they change the protein, and that protein has a function, and that aberration in its function will result in the disease, basically. But we also have non-coding variants that are disease associated, that are called pathogenic, basically. Pathogenic non-coding variants. Non-coding variant, it means that there's a change in the genome relative to the rest of the population on average, that doesn't actually code for anything. It's not part of the protein. It's just in the middle of nowhere. A lot of these are actually intronic, right? So they're in an intron, so they don't really impact the protein at all. But what they do change is splicing, potentially, right? So we can actually take advantage of our model to measure what the impact of this change would be, right? So if this is the what we call the reference isoform, so there's in this position, there's a G, and then there's a mutant isoform where there's a U there. You can compare the likelihood of these two isoforms using our model. And the more the two isoforms the likelihoods are different, the more likely that this change actually has an impact on the RNA structure, architecture, right? And as a result of that, it's more likely to be pathogenic and disease causing. So there is this nice data set of clinical variants, which is called ClinVar. You can subset it for non-coding variants and basically run this analysis that I just said. Compare the difference in likelihood of the reference versus mutant versions. And now this is a classification task because we know some of these variants are pathogenic, some of them are not. This is again comes from actual clinical data, so we have the ground truth there, and you can actually ask how well does my model capture, basically using this delta likelihood as a metric, as a zero-shot metric of the impact of these variations, genetic variations, how well does it capture what would be pathogenic and what would not. And again, you can see our model outperforms all the other models, including a Splice AI, which again is I would consider the state of the art. Okay, so these are basically just using likelihood from the model as this to look at this zero shot tasks. So the model is clearly understanding the transcriptome architecture, right? It's very clearly capturing something about the architecture. But what comes out of the model is just not likelihoods. You also can get the embeddings for each of the tokens. So you can actually look at the end of the isoform and get the embedding of that isoform. And if you do a 2D projection of it, actually looks something like this. And you can ask, like, why are the sequence differences that clusters them differently, and you'll get enrichment of different sequence elements. And these are the barcodes that I was talking about. So again, what makes some like an isoform more or less likely is dependent on this barcodes, these sequence barcodes that it carries within it. That actually determines what happens to that isoform, or how likely is it that isoform for it to be spliced into. And our model basically has learned that. And the point is that if the model can predict that, then it's also leveraging these same barcodes. Right? So let's ask that question. Does it actually do that? So first of all, as I mentioned, we started with 26 cell lines, right? So far I just averaged them. As I said, you can average them all as to get like a universal measure of transcript abundance, but there are of course individual variations as well. Uh, but if there is this barcodes embedded in the transcriptome that governs how a splicing happens, what it means is that if I know the identity of these barcodes for a subset of the transcripts, I should be able to predict the rest of them. And that's one assumption, which is actually a good assumption. So if I learned what these barcodes are for like a subset of what I'm showing you here, for the subset of the transcriptome, then the rest of it I should be able to predict um, based on the presence or absence of these barcodes. That's one thing. The other thing is that our other assumption is that our embedding actually has learned these barcodes. So within it has captured these underlying barcodes. So the way to test this is, can we predict isoform abundance? Now is a few shot regime basically, using the embedding of this final token. So can we use but this final token is called polyacide pass. So it's just the last token of the isoform. So can we use the pass token embeddings to predict isoform abundance? So we train on a subset of isoforms. We take for every isoform, we take the pass token from it. It's basically a regression task. So we train on a subset of isoforms and we test on the other subset. And as you can see, our model actually does pretty well across all these cell line models. And you can see one of them, an example here with a 0.5 correlation. So very nicely, you can learn the relationship between the embedding, the pass token embedding, and the isoform abundance, which, as I mentioned, basically highlights the fact that those assumptions that I told you are likely true, right? So the model actually has learned these sequence barcodes of that regulate its splicing. Another way that you can ask a similar question is that do a pairwise comparison between cell lines, and there are specific isoforms that are only appearing in a given cell line and not the other, right? And again, the idea is that there is an underlying grammar that is driving this. So there's like one or two pathways, one or two kind of regulatory mechanisms kicked in one of these cell lines that is driving the changes, right? And if I know it, then again, if I know a subset of these like cell line specific isoforms, I should be able to predict the rest of them, right? So now this is a classification task. So the accuracy of your classification task as measured by a UROC in this case. So it's a pairwise task. So for every pair of cell lines, it's also not symmetric. So for every pair of cell lines, we say what is specific to this one that is not seen in this one. And if I know a subset of them, can I train a classifier, again, using the token embeddings to predict the rest of them? And again, you can see we do a pretty good job of that. So again, the grammar of splicing is summarized in the token embeddings that the model is providing. So now the final step in the final few minutes gets to even further really understanding what these sequences are. I mean, again, I'm showing you that the model understands this. It's probably like we talked about barcodes. They're probably somewhere in the sequence, but where are they, right? And how did the model learn them, and where are they, and how can we find what the model has actually learned? So again, the model is learning these barcodes, and it's summarizing the past token embedding, but that doesn't give me as a biologist an understanding of splicing at all, right? So we need to be able to link that embedding back to the sequence space, which I can understand, right? So let me give you an example, like a real world example that would actually let us double click on this a bit. So this is a very famous, gene. It has two variants. So one, you see this exon in the middle. This exon is either included or not, right? So these are the two splice sites of the exon. And we know from previous work that this decision is driven by these two parts of the intron where a protein, RNA binding protein called RBFOX1 sits. So if basically RBFOX1 binds these places, And if it binds, this exon will be included. If it doesn't, it won't. So if you remove RBFOX1 from the cell, you won't see this exon in the middle. Right? So this is something that we do know about biology, right? So does our model understand this? So does our model understand all of these important points? So does our model understand these splice sites? So splice sites are actually very defined. They start from AG, end with GU. And they have other signals as well, but these two are the most important parts of it. So as now, the way that we can govern this is actually look at the basically the model likelihoods again, right? So how sensitive the model is to mutations or masking of these positions across the sequence. Now we have two isoforms of structures. One where this exon is not included, so it's treated as an intron, which is here down in the bottom, purple. You see that the model, when this is an intron, the model doesn't care at all. Like nothing matters. Like you make your changes, you mask all of these tokens, and nothing happens. But when this is included as an intron, as an exon, so here in this row, then the model actually pays attention to things that should matter, right? So for example, the splice site AG, if you mask it, the model likelihood changes a lot. Here as well on GU, changes a lot. So the model has understood what the splicing signals are. This is not too hard, as I said. It's a nice AGGU. But now, does it actually understand these RBFOX1 sites as well? The answer is yes. As you can see, the red parts are where RBFOX1, known binding sites of RBFOX1. And again, as you see, if you mask these tokens, the model likelihood changes. So the model understands that has linked the presence of this exon to the presence of this signal. So again, this is what I was calling barcode. So this is a barcode for RBFOX1. And the model has actually learned that RBFOX1 is a regulator of splicing. So that was one example. Now we can do it kind of transcriptome wide. And so what I've done here is basically focused on the splice sites. These are called splice site acceptor and donors. The two ends of it, doesn't matter what the name are. But the point is that across the transcriptome, if you mask or mutate these splice sites, the model likelihood changes, right? So the model has really internalized the splice site donor acceptor signals, which is really nice. Now, going beyond this splice sites, how do we find other barcodes, right? So barcodes like this, right? Like RBFOX1, for example, that are not exactly in the splice sites, they're in the other parts of the transcript. So we do this in two ways. One is going off of what is known. So for example, known RNA binding proteins, knowing where they sit and asking whether that position matters for splicing, or getting a sliding window, taking it across the transcriptome, and just ask, if I mask this part, what happens? If I mask that part, what happens? Just basically do a kind of a systematic search of that. So first of all, we went with a database of known RNA binding protein binding sites. These are all the RNA binding protein listed here. It doesn't really matter what they are. But you can sort them based on how much the model thinks each of these binding sites will impact, each of their binding sites impact the splicing, right? So you can sort them based on that fact. And if you do that, you actually see that there's a very strong enrichment of known splicing factors among those binding sites that are overreacting, right? So that's a good sign. Again, another validation that the model had really understood that the binding sites of these splicing factors is what drives the splicing, which is great. But some of these are also new, because you can tell even for a given splicing factor is not always a splicing factor. Sometimes it is, sometimes it's not in different contexts. So the model has actually learned this as well. And finally, as I mentioned, you can also do this systematically by just having a sliding window that goes across the sequence and asking what happens if I mask this piece. And out of this comes out sequence barcodes that are important. So, and again, now you can go post hoc and assign them to known RNA binding proteins. Again, like for example, this sequence that we find very important, our model thinks it's very important for splicing. Turns out it's very similar to what SRSF1 usually binds, and SRSF1 is known splicing factors. But again, I'm like summarizing everything, but the point is that you can actually go deep, and for every isoform in the transcriptome, understand like what parts of this transcriptome LoRNA thinks are important for the architecture. And this will allow you to fix problems in the splicing. There are splicing diseases that you can actually you would be able to fix this way. You can learn how to fix it. What are the proteins that are actually driving that effect that you need to modulate to be able to fix it, and so on and so forth. And a final minute, LoRNA is a generative model. It's a causal language modeling approach. So you can actually use it to generate sequences. So we actually wanted to also ask this question of, if you use LoRNA to just create synthetic RNAs, what would that look like, right? So we are giving it the prompt, which is human and the start, and then it gives you like a series of exons and introns until the very end. So when we looked at, and then we looked at, the RNAs that the genes, basically the isoforms that the model is predicting, we see that the exon length is actually pretty good. The distributions are pretty similar to what we see in the human genome. Intron lengths, again, pretty good, not that different. Number of exons per transcripts are pretty good. Sequence length is decent. And also, if we take the sequences that we have generated and give it to a Splice AI, which is the other model of splicing, actually for the vast majority of them, the Splice AI thinks that, yeah, there are like legit splicing events, right? So this is the likelihood of Splice AI. Now, in all of this, like the model has learned nothing about exons and coding sequence and proteins or anything like that. But it turns out it also learned that as well. So in these synthetic sequences, if you look at the codons are basically these 3 letters that code for proteins for each of the amino acids. So you have 20 amino acids, but 64 codons, right? So each of them codes for a different amino acid, but one amino acid has more than one codon. But they're not uniformly distributed. And so every organism actually has a different distribution of these codon frequencies. And this is called codon usage. So human has its own codon usage, for example, which is different from mouse. But if you basically compare the human codon sequence, codon usage in our human coding sequences, basically, it's very correlated with this synthetic codon usages from LoRNA. So LoRNA is creating this entire transcriptome, which like a lot of is even intron, and like has seen very little exon, and never actually exons is stitched together to be able to understand that this is a contiguous coding sequence. But it actually learned and internalized this coding abundances as well. So again, all of this to point at that, this model does incredibly well. It has learned a lot about the transcriptome architecture. It can be applied zero shot or few shot to answer like key questions about the splicing as like a key step in gene regulation in humans, and finally be able to use to dissect biology to understand what actually drives this process. So you can do like trillions of in silico experiments on splicing, whereas these actually doing them in the lab is quite difficult. As I mentioned, we have the preprint out already. If you want to go check it out, by all means do. We published this a few weeks ago, and the code and everything is also published as well. And again, the folks who did this, Ali and Benedict, were the 2 lead authors, and this was a very close collaboration with 2 of my colleagues at McGill, Hamid and Amin, and Vijay Ramani at Gladstone Institute here in the Bay Area. And I'm happy to take final minute any questions that you guys might have.
Cool. Yeah, thank you very much. Folks in the call, do you have any questions?
Yeah, great talk. Thank you. I was curious. I have a number of questions, but I'll try and limit, I guess. I was curious, did you look at all at varying the context length, how much benefit you're getting? I know like Hyena DNA, they say we can get up to 1 million base pairs context length. And so certainly you have Titan and, other very large genes, and how much you think you'll gain by extending beyond 64K.
Yeah, I mean, so the question is how many genes are in the end above 64K, and if you kind of lengthen the context, kind of how much data do you need to be able to train that nicely, and also how stable the training will be. The StripedHyena architecture, the code is becoming more and more stable, so we can certainly increase, and that's part of the plan. But 64K, I think it's like more than 85% of the genes fits in that, so it's a good number to be in.
Gotcha.
But certainly can be increased, yeah.
In the you mentioned at the end the synthetic generation that motivates a decoder-only model. I'm curious for prediction of splicing, like how much you think you're losing by dropping 3' interactions by having just pure causal attention.
Very good question. I mean, we have always been kind of surprised for how well this causal language modeling as opposed to math language modeling actually works in biology. It's less surprising in case of RNA because splicing is actually co-transcriptional. So splicing happens as its RNA is being transcribed. So actually, all of the RNA doesn't even exist necessarily. So there is some gap. So at least in case of splicing, you could like rationalize why like one-sided causal language modeling can do well, partly because of this co-transcriptional approach. But for some reason, even genome language modeling, like the EVO model out of ARC, really worked well, despite the fact that it's one-sided. I mean, there have been like hacky versions of it for high enough for two-sidedness, but that doesn't really work. It just breaks causal language modeling. So the short answer is my sense is because of redundancy, that you have like codes that are redundant in the genome and transcriptome. So even if you're missing like one side of it, there's enough information on the other side to be able to predict what will actually happen.
Awesome, thank you.
Thanks.
Sure.
Maybe one more thing. It sounds like we're very much in the process of getting better equipment to get longer reads, higher throughput. So how will that affect the kinds of data you have in terms of having time courses, diversity of cell lines, or where can this work go?
So basically, I mean, you're always data starved in this, kind of in this regime, right? So more data helps you build better models to do more better in silico experiments, right? So for example, here, yeah, it took us, a ton of money and a lot of effort to be able to just look at baseline expression of these isoforms in like 26 samples or whatever, like 250 samples. But like, for example, if you want to go and like take all the splicing factors, like modulate their activity and measure their consequences, like these are all, they become very interactable very quickly, right? Whereas you can do easily all of that in silico, right? To understand like what parts of the transcriptome matter and where to focus on and what to study. So all of I mean, in biology, we really care less about the performance of models and care more about like understanding, like using the model as a way of dissecting biology and understanding what it's learning, right? So that we can do experimentation in silico as opposed to in the lab where things are a lot slower and a lot more expensive and not very scalable.
Right. Well, yeah, thank you very much. It was fantastic.
Awesome. Thanks for having me. It was a pleasure. And please do reach out if you have any questions. I'm one email away, and have a nice rest of the day.
Yeah, you too.
Thank you all.